Join the conversation.
Join the conversation.
You're with Cape Talk.
And you've been waiting for this time, our weekly interaction with the Naked Scientist, consultant virologist at Addenbrooke's Hospital in Cambridge, Dr. Chris Smith, every Friday just after 9:30. Welcome, Chris. It is exactly 9:35.
Good morning, Clarence. How are you?
I'm good, thank you. Absolutely wonderful. And you?
Yeah, I'm not doing too bad. I've had quite an interesting week on the science front. Had some interesting interviews. You'll be able to catch a couple of them on the Naked Scientist podcast that we're going to publish later today. But one of them, I spoke to one of the team last night behind the mission in 2022 to slam an impactor into an asteroid. Do you remember this? The DART mission. And this summoned up visions of Hollywood and deflecting asteroids. But that really is the premise of the mission, to understand if there was something racing towards Earth at 10 kilometres a second, which is about the speed these things go at, and it was going to slam into us, could we, with enough notice, deflect it? And the way that NASA and other scientists approached this was they found this asteroid. It's actually an asteroid with a little moon going around it. It's called Didymos, orbited by Dimorphos. And what they did was to send a probe to it and then slam a big impactor into the little moon, which measures about, I don't know, a few hundred metres, 150, 200 metres across. Very, very small, but just about held in the gravity of the bigger body. And they were able to see what happens when you do this, see how the orbital period of this little satellite going around the asteroid changed. They made it change by half an hour. It went from 12 hours of orbit time to 11 and a half. But they've since done more follow-up, which is what they're publishing this week, and what I was talking to Steve Chelsea, one of the lead researchers, about, which is to look at how that effect has changed the trajectory of the whole thing going around the sun. And in fact, they were able to get amateur astronomers to make a measurement to within a precision and accuracy of seeing a human hair at a distance of 20 miles. That's how good the amateur astronomers with their telescopes on the ground were when they made these measurements. And they can see how much of a difference they've been able to make with this mission to the orbit. This was obviously just a test, a proof of concept, but they were able to move the whole thing. And they said this is the first time that humans have moved a celestial object in space in this way. So we now have a much richer understanding of how we can deflect asteroids, by how much, what it takes, and what happens actually when you do this, because it's not as simple as just slamming a big lump of copper into a heavy rock, because these asteroids are actually made of lots of bits of rubble and material, and when you slam things into them, they actually disintegrate, and you get a kick from all the stuff being ejected off the surface, which also changes the maths a bit, and they've been able to resolve that with this new paper. Really fascinating. You can hear Steve Chelsea talk about it, as I say, on the Naked Scientist podcast that will come out later today.
That's interesting stuff that I'll go and find. Olive, you've beaten Shireen because you called in and we prioritize the calls. Go ahead with your question for the Naked Scientist.
Hi, good morning. I would like to know why you can't tickle yourself.
Oliver, good morning. Let's have a go together. Well, I'm trying desperately to tickle myself, and I'm not succeeding. You're quite right. And if I had someone sitting next to me, though, and I quickly reached over and I sort of put my fingers, with their permission, of course, just above their hips and went squeeze, 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 they would probably climb the wall. Why is there that difference? Exactly the same movement, but done to a third party drives people crazy. Do it to yourself. Doesn't make any difference. We think the reason for this is that there is a region in the brain, and it's somewhere at the junction between the parietal lobe, which is the bit, if you put your hands on your head as if to say, oh my goodness, the bit at the back, the occipital lobe, and the bit at the side, the temporal lobe. Where those three bits of the brain meet is the temporoparietal occipital junction. And in this region seems to be a cluster of nerve cells whose job it is to compare what you think you're about to do with what is coming back in from your body, and anticipating what signals will come in and restricting or subtracting them from effectively what is presented as novel to your consciousness. And the reason researchers are even interested in this bit of the brain is because it may go wrong in people who suffer from things like schizophrenia, where they're subject to distressing hallucinations. Because in a hallucination, we're effectively feeling something or seeing something or hearing something. It's coming from our own brain, but we don't realise our own brain made it because something is wrong with that subtraction process. I haven't asked someone with schizophrenia if they can successfully tickle themselves, but you would anticipate they ought to be able to surprise themselves a bit more than someone who doesn't have schizophrenia. So we think that the reason you can't tickle yourself or fool yourself in other ways is because you have this brain centre comparing what you're about to do, anticipating what that effect will have on the body in terms of the flow of information back into your brain, and then subtracting the two. And because you're expecting it, it's not a surprise and it doesn't drive you crazy.
Shireen came through with the first question at 7 minutes past 9 o'clock. A question for the naked scientist. It reads, how does having a low lung capacity affect the heart? I'm a non-smoker, says Shireen.
Hi, Shireen. Well, lung capacity varies between humans. To an extent, we can train it or improve it, but some people are born with a bigger set of lungs than others. It's just part of natural variation in the same way that we vary in height. On average, men have a bigger set of lungs than women, but there are some women who have very big lungs. And through the training effects that you can achieve, you can also make that bigger. And so women who are very good at synchronized swimming or free diving also tend to have a bigger lung capacity. By lung capacity, we just mean how much air you can put into your chest cavity. Your lungs are effectively sponges which are hanging in your chest, and when you raise your chest wall and lower your diaphragm, the pressure in your chest cavity becomes lower than the pressure in the atmosphere around you. So atmospheric pressure pushes air into your lungs, and it keeps going until the pressures equalise. And when you breathe out, you reverse that process, and the elastic tissue in the lungs recoils, your chest wall falls, your diaphragm comes up. It puts the pressure in your chest up above atmospheric pressure again, and there's a net flow of air out. And what's left behind we call the residual volume. This is the amount of air that you can't push out of your lungs because you'd have to create even more pressure in your chest than you actually can, and therefore there's always a residual volume left behind. So there's lots of different things that we mean by lung capacity, but really we're talking about how much you can breathe in and out in the way that I just explained, and that's down to how much lung tissue you have, and the bigger your lungs, the more you can move in and out. This does not have an effect on the heart in health, because when you breathe in, you lower the pressure in your lungs, you make it easier for blood to flow through them. That's an effect. And when you breathe out, you increase the pressure, pushing blood back into the other side of your heart, so that you actually transiently increase your cardiac output, actually. But it doesn't have an effect day to day on a healthy, normally working heart.
Maybe just, it's a loaded question from Roger. I don't know. You tell us, Dr. Chris Smith, the naked scientist, and keep your questions coming via WhatsApp at 072 567 1567, and you'll jump the queue if you call and put that question to him directly. 021 446 0567. If a white person's skin is transplanted onto a black burn victim, will it remain white, or will it change to the recipient's shade over time?
This could be revoiced as, or reworded as, if I took a liver from a white person and put it into a black person, will I get a black person's liver? It doesn't matter. Skin is an organ, blood's an organ, livers and kidneys are organs. The reason that a set of lungs put into a person with cystic fibrosis from a person who doesn't have cystic fibrosis cures the lung problem in the person with cystic fibrosis. The reason a bone marrow transplant from a person who doesn't have a bone marrow type cancer put into a person with a bone marrow type cancer can cure the bone marrow type cancer person is because the cells that you put in are healthy, the cells in the organ you took out were not. So you don't replace the cells in the new organ. And as a result of this, you effectively, if you have a bone marrow transplant, you've got a person who's your donor's DNA going around in your bloodstream. You become a chimera, a mixture of them and you. And in fact, some people will say, well, my blood group changed after a bone marrow transplant. So in other words, you need the cells from the donor, because they're the healthy ones, to help you to recover. So with a skin transplant, the reason skin that is black is darker is because although the number of melanocytes is exactly the same in both white skin and black skin, melanocytes are the skin cells that make pigment, they're just more active in black skin than white skin. If you put full thickness skin transplant, black skin onto a white person and vice versa, you'd still end up with either a black patch or a white patch, because the cells, assuming the immune system was controlled and didn't reject them, would still be there, just like that bone marrow transplant and just like that liver transplant or kidney transplant. So therefore the melanocytes would still be the same. They're from the donor. They would have the same level of activity, and therefore the skin would have the same tone as the original donor.
Zuki, follow-up question on the tickle question of earlier. Welcome, Zuki. Go ahead for the Naked Scientist.
Okay. Hi, Dr. Chris. Yeah, I was just interested. Why is it that some people aren't ticklish, especially in that region above the hip? So basically, what determines how ticklish a person is?
Well, morning, Zuki. Tickle is relatively poorly understood. We know that we have a population of nerve cells that sense itch, and we also have a population of nerve cells that are called low-threshold mechanoreceptors that seem to convey tickle sensation. Part of their role is to alert us to when something's trying to drill holes in our skin like a parasite and get in and cause us problems. And that's why we pay so much attention to itch and tickle. But one of the other important things to bear in mind with tickling, and when you have the sensation of hands on hips or fingers either side of the knees and squeeze, everyone knows what that's all about. Really, really uncomfortable and drives you crazy. That's called gargalysis, is the sensation that it elicits. And it's not sort of a coincidence that young children spend a lot of time with their parents being tickled. Other non-human primates will tickle each other. Other animals will do this. Where are the tickly bits? They're your vulnerable bits. You think your hips and tummy, your knees, under your arms, all the places where you're particularly vulnerable. And so when we tickle, what we're actually doing is alerting a younger person through play to where their vulnerable bits are. And what do? Well, they learn very crafty techniques to roll out of the way, deflect, dodge, jump back, jump forward, effectively protect your vulnerable bits by making it fun to learn where they are. So scientists speculate that we have this mechanism in the first place as an educative thing, but not everyone is the same. And some people are going to have regions of the body where we're more sensitive than others. Some people are better at suppressing this sensation than others and controlling themselves. And I think it's probably a combination of all of those things why some people are more ticklish in some places than others.
Gisela has sent us a voice note. Let's take a listen.
Can you tell us a little bit more about ancient DNA, which apparently they had found in Greenland, in the northern parts of Greenland, that was, I think, 2 million years old? Do you know about it? It was a very interesting blog that I saw on YouTube.
Hello, Gisela. I don't know the particular story you're referring to, and if you'd like to send me a reference to it, I can take a look and I can then come back and report a bit more in a subsequent week. You can write to me. It's chris@thenakedscientist.com. But certainly I can talk more generally about ancient DNA, and that is that we used to think that the living, acting us, once we died, everything broke down and there was not much left, except if you made a fossil, and that was just a stony replica of what had once been there. But in recent years, we've realised that there's a lot more to a fossil, or a lot more to material, than we had believed. And in fact, there are some of the molecules left behind that were part of that entity when it was alive. And this includes plant tissue, animal tissue, even human ancestor tissue. And we've got much, much better now at getting some of those molecules out. In fact, there was a lovely paper published about 10, 15 years ago now of a mosquito fossil found in what would once have been the mud at the bottom of a shallow pond at the time the dinosaurs were knocking around, so from about 65 million years ago, and they could still see hemoglobin, iron from the hemoglobin of that mosquito's last blood meal in the back end of the mosquito using clever X-ray techniques. But in terms of DNA, DNA is just another molecule that is in almost all the cells in a living thing. And it turns out that DNA is a bit more robust than we had first anticipated, and it doesn't just break down and disappear, and we can get the DNA out. Now, the challenge is that the DNA in you and me, in each of our cells, there's about 2 meters of it in a big long string, wound up like spools, and they're the chromosomes. When we're living, there is a mechanism at play to read the DNA, check the integrity of the DNA, and repair the DNA so that, for most of our lives, it works perfectly. Obviously, once we die, we no longer have that mechanism anymore, so the DNA is then subject to all of the damage that can come in from the environment, drying out, temperature changes, degradation from microorganisms, etc. And this has the effect of fragmenting the DNA into short pieces. Now, scientists originally thought that was a showstopper, but we've got sufficiently good now that with enough samples, you can read the little bits of DNA and then get very good computer programs that will do the equivalent of a multi-billion piece jigsaw puzzle to work out how all these bits overlap, because lots of the fragments all overlap with each other, and you can then work out what the original sequence was. This has been applied to Neanderthals, which haven't walked on Earth for tens of thousands of years. So we have the full Neanderthal genome, and that won someone a Nobel Prize, so we can compare Neanderthals with our species. We've also got ancient organisms. S.K. Willerslev, who was from Copenhagen originally, I think he's now based in Cambridge, had the record for a while, at least, for doing a horse or something from about 750,000 years ago. So all the time, this is getting better and better, our ability to get these molecules out of ancient specimens and then ask clever computer programs to piece them back together. We're getting better and better at that. So the possibility of getting DNA from things that are a couple of million years old doesn't surprise me in the slightest. But do send me the story. I'll take a look.
It is 9 minutes to 10 o'clock. The Naked Scientist with us through till 10 o'clock. And keep your questions coming through for Dr. Chris Smith via WhatsApp 072 567 1567. Peter, you have jumped the queue. Welcome.
Hi, good day. Dr. Smith, I'd just like to know, why is the sea salty?
Hello, Peter. The answer to this one is that it's all about the water cycle. The Earth has got a huge amount of water on it, most of it in the oceans, and that's being heated by the sun. Each square metre of the ocean surface is getting hit by sunlight at the rate of about 1 kilowatt. So it's like you having a 1 kilowatt heater blowing on each square metre of the sea surface. This gives enough energy to some water molecules at random to break the bonds from the water molecules around them and, you've guessed it, evaporate. So you have water vapour. But salty particles, because they're electrically charged, can't leave the water surface in the same way, so they're stuck behind. So what evaporates from the ocean is clean, pure water, as water vapour in the atmosphere. This is warm, and so it rises as less dense air rises, and it goes up in the air, cooling and expanding as it does so, until it gets to a point where the energy is sufficiently low that the water molecules, instead of being spread out, come back together, join up, link up, and make water molecule groups. We call them raindrops. So you get clouds. This blows inland, over land, and you've again guessed it, you get rain. Down comes the rain. This is pure water at this point, picking up a few things from the atmosphere on the way, like carbonic acid and so on. It hits the ground, and it then waters our gardens. As it percolates through the ground, though, it's going to pick up traces of salt and other particles and dissolve minerals from rocks. And that carbonic acid I mentioned is going to help it to dissolve things like limestone, loads of that around Joburg, for example. And as this water then collects into streams, it's now no longer pure water. It's water with a small burden of salts and minerals dissolved in it. This flows back to the sea, and you can see we're getting back to where we started, because it takes with it that small salt burden and dumps those salts in the sea, and then the process starts again. But this time, the water evaporates, leaving behind those salts that it picked up from the land in the first place on that journey. And if you keep running this cycle for millions and millions and millions of years, you're slowly adding salts to the sea and putting fresh water on the land, and the sea salt concentration is therefore going to rise up and up and up until it reaches a threshold point where if you add any more salt, just like trying to stir sugar into your tea, you get to a point where you can't make any more dissolve. You get to a point where any more salt added to the ocean begins to precipitate out, forming minerals that coat stuff on the sea bottom. So you end up with a roughly steady state saltiness in the sea and fresh water on land. Thank goodness, otherwise we'd all be drinking salt water.
And Kin writes in response, great and detailed explanation of the water cycle. How does one prevent the onset of seizures? I just found it happening recently at age 55.
It depends why a person is having seizures. Seizures are fits. They're also sometimes called epilepsy, because if you get a run of fits and they're originating from one part of the brain and they're part of a syndrome, you may well have epilepsy. But a person who has a head injury, a person who has a problem with an infection, they can also cause fits. So a fit is an electronic discharge in the brain, usually involving the whole brain where the nerve firing sequence has gone off kilter, and as a result, consciousness is affected. So the first thing to do with this is to ask, well, why is this happening? Things don't happen for no reason. Has there been a brain injury? Is there something in the brain that shouldn't be there, like a tumour, which doesn't have to be cancerous? It could be a benign tumour, something's pressing on the brain. Has there been some change in drugs or medication that has caused irritation or damage to the brain? Has there been an infection earlier in life which has caused a problem in the brain? There's an infection that you can pick up from pork called cysticercosis. It's a worm. It can embed itself in brain tissue, and when the worm dies, it can unfortunately trigger the brain to start causing seizures. Various reasons. There was a lovely paper actually by researchers from the University of Cape Town who studied this and published it in the journal eLife in the last year or so. So have a look at that, because they explain in great detail how these worms getting into the brain are having a chemical conversation with brain cells, possibly to make space for themselves in the first place. But when they die, that's when you have a problem. So the first thing we would do is, if a person suddenly started having fits, is to confirm they're really having fits. And you can do that by measuring the electrical activity. You then need to have a look at the structure of the brain to see where this might be originating from. You get some clues from the symptoms, because some parts of the brain tend to produce certain symptoms or certain patterns of fits, and so we can learn something from that. The brain scan might point to a cause, and then that cause will dictate how we intervene and treat the problem. And if it's a damaged cluster of nerve cells, there are lots of drugs, some of which are more tolerable than others, many of which are very, very good and don't produce lots of side effects, and they can be used to damp down the neurological activity that's causing the fits. In other cases, it might be necessary to go a stage further and have surgery, even to remove bits of the brain that might be causing the problem in the first place. So really, when a person has fits, there has to be a reason for that. It has to be investigated, and on the basis of what is found, there are a range of managements which are more or less invasive, which are tailored to why that person has that problem.
We're going to have to wrap it there. It's 2 minutes to 10 o'clock. I'm just amazed how time flies in this little half hour that we have with Dr. Chris Smith, our naked scientist, every Friday. Thank you, Chris. It's appreciated, and looking forward to next week already.